next speaker is Dr. Craig Venter. UCSD always takes sort of credit for Craig since he received his PhD in the physiology pharmacology graduate program here with the late Nate Kaplan as his mentor. Craig, I remember, went directly to a faculty position. He finessed a postdoc. That was, I think, a pattern of his rapid movement in whatever he did. He then moved to the NIH, where he developed the express sequence tag methods for rapid DNA sequencing and powerful computational methods to link sequences in decoding the human genome. Craig founded the Institute for Genomic Research, which decoded the first genome of a whole organism, a Haemophilus influenza, and he founded Celera Genomics, and as they say, the rest is history. He is now president and chairman of the Center for Advanced Genomics, which is using high-throughput technologies to link genotype to phenotype in human disease. So his talk is entitled Advancing the Revolution Through Genomic-based Medicine. Craig, welcome.
Thanks very much, Gordon. It's a pleasure to be briefly in La Jolla this morning before I have to run back to the cold East Coast, particularly for a sailing trip. Looking back at the last 50 years, I'd say they'd be best characterized by a function-to-sequence approach, at least 70% of that time. The last 15 years have really been focusing much more on sequence-to-function. The sequence-to-function actually started in 1978 with Fred Sanger when he sequenced the first DNA and the first genome. It was actually bacteriophage Phi-X174. That I'll come back to later in my talk. I'm going to tell you about some of the work of the 400-plus scientists and staff we have in our three basic science institutes, TIGER, the Center for the Advancement of Genomics, and the Environmental Institute, the Institute for Biological Energy Alternatives. These three institutes have a high requirement for high-throughput DNA sequencing, and we recently invested $50 million in building a new high-throughput sequencing facility that does a little over 100 million base pairs every 24 hours. These institutes have now over $100 million a year budget, basically all with competitive grants and contracts, but the front edge research is funded by our foundation. Whole genome shotgun sequencing took off in 1995 with the first genome of a free living organism, Haemophilus. And as of a few months ago, there's now over 150 genomes that have been completed. But this exponential pace is continuing to grow. And it's likely by the end of 2005, when the first draft of the human genome was supposed to be sequenced, we're going to have well over 1,000 genomes sequenced. The first two that were done in 1995, Haemophilus and Mycoplasma genitalium, barely exceeded a few million letters of genetic code. Last year, multiple institutions did in the hundreds of billions of letters of genetic code. We scaled up things further in 1999 and 2000 at Celera with sequencing the Drosophila genome in collaboration with Jerry Rubin. This took a 10-year project and did it in 4 months. And as soon as Drosophila was finished, we started it on human. We sequenced the genome of 5 individuals, 3 women and 2 men of self-described ethnicity as African American, Hispanic, Chinese, and Caucasian. And using the paired end strategy and the algorithm with a team headed up by Gene Myers, we covered the genome 39 times in a 9-month period, published our results in Science at the same time the public consortium did in Nature. This is where the surprises begin to come because we found far fewer genes than most expected. Some in the biotech industry were expecting hundreds of thousands of genes. Bill Hazeltine had patented 300,000 of them, which says something about the patent office as well. Insight jumped up and said they had 375,000 they patented. The reason there were so few in our understanding is we didn't know the degree of heterogeneity in the genome, and that we had most of the genome in deserts with millions of letters of genetic code with few or no genes. We're just finishing the analysis of a number of genomes now that the NCBI has built a relatively complete, quote, final genome sequence. We can start to retrospectively analyze what was done. This is a Celera-only whole genome shotgun with the data generated early on at Celera that will be published shortly, comparing what was published early on, finding a lot of disjointed things, but as the NCBI builds improved, they approached the mathematical assembly, showing the value of mathematical tools in this field. As soon as we finished human, we started on mouse, as did the public consortium, and I think that's where things started to get interesting. Finding all the genes in the same order in human chromosomes and regions of mouse chromosomes. For example, here's mouse chromosome 16. Here's this region from human chromosome 21, basically one line for each gene. You can see gene order and content being highly conserved. We found 116 genes on mouse chromosome 16 that did not occur in human, which was some relief to some people. We recently published the dog genome, and things got even more interesting. This is Shadow, one of our standard poodles, who took time off from running a DNA sequencer to give a blood sample. You heard they could be run by monkeys. Actually, they can be run by standard poodles. The dog genome is interesting because the degree of phenotypic differences all derive from the same wolf genome, over 6-fold in height, 30-fold in weight. But I think one of the most interesting things going forward in this field is the distinctive behaviors and diseases associated with different dog breeds that I think the dog genome will probably help more than any other genome to help us deconvolute the genetic components of behavior. Here's a series of genetic disorders in dogs. Most of these have corresponding ones in humans. And just to give you a couple examples of the segregation, deafness occurs in about 10% of Dalmatians, von Willebrand's disease, and about 50% of Doberman Pinschers. Just since we published this data, a major form of eye disease in dogs was discovered and the gene discovered, and there was a human correspondence to it that has now made some very nice progress very quickly. I think the biggest surprise with the dog genome was that it was closer evolutionary to humans than the mouse. If we take all the mouse sequences and line them up on the human genome, we get around 378 or so million letters of genetic code that align almost perfectly with our genome. We do that with the dog sequence, even just at 1.5x coverage, we get over 640 million letters of genetic code aligned. That's about half of the euchromatic portion of the human genome that corresponds tightly to the dog genome, not just in gene content and order, but much broader. These studies show what was beginning to come out of looking at the mouse genome, that it's clearly evolving much faster than dog or human. In fact, where dog and human are much closer to whatever the common ancestor was, changing our view of genetics. When we look at the synteny, we see it over much greater regions of the chromosome with gene content and gene order. As you heard from Dr. Sakaki, obviously we're even closer to chimpanzees. This is Pabo's data showing the human X chromosome differs only 0.9% from the chimpanzee X chromosome, which I've argued proves that the shopping gene can't be on the X chromosome. Also, the Y chromosome with very few genes on it appears to vary quite dramatically between humans and chimps with no impact on behavior at all. But many of you already knew that. Lee talked about human-based genomic medicine. Right now, we're in the era of gene-based medicine with prenatal testing, and I agree with him and others that we're going to move towards genomic medicine based on having the complete genome of individuals. We need a lot of development still. We need the kind of technology development he talked about in the genome field. My foundation at the Genome Sequencing Conference announced a $500,000 prize to whichever scientists come up with the technology that leads to the $1,000 human genome. We need to learn how to interpret this information and get digitized clinical records, and we need a dramatic new computational infrastructure. I've argued the doctor-patient relationship will go largely unchanged, except that it's going to be consumer-driven as each of us are able to get access to our genetic code, to get access to the internet. Most people right now, when they hear they have a disease or somebody in their family does, you go to the internet, and most of us end up educating our physicians, not the other way around. We're working with groups both here at UCSD and at Duke on a preventative medicine paradigm, particularly with Ralph Snyderman, who's been a major advocate of this at Duke. And a great example is with the DNA repair enzymes in non-polyposis colon cancer. Early on with the EST method, we found 3 new DNA repair enzymes that Bert Vogelstein showed were linked to colon cancer. And there's now commercial tests available to get your DNA repair enzyme sequenced. They won't tell you whether or not you'll get colon cancer. They'll tell you whether you have an increased probability or increased risk of disease. But I argue that's extremely valuable information. Right now, the medical practice says to get a colonoscopy when you turn 50. A lot of people die from colon cancer long before the age of 50. If you know early on that you have an increased risk of cancer, you can do something about perhaps changing your diet, certainly getting increased physical exams. The statistics are pretty overwhelming. If colon cancer is detected after symptoms appear, there's about a 65% chance of a 5-year survival. If the colon cancer is detected early before there's symptoms, there's greater than a 95% chance of a 10-year survival. That's a pretty substantial difference that we can put that power into individuals' hands instead of waiting for random events. There's increasing studies for all over. This is one from Nevin's group at Duke on looking at metagene analysis, gene expression in tumors to predict outcomes. This is one with breast cancer. It's pretty overwhelming, but we've seen now dozens and dozens of studies like this from around the world, all with very similar outcomes. Patients can be divided into high and low risk group. In this case, ones for developing nodal tumors after the primary tumor. They were divided initially into a low risk and a high risk group. But Nevin's group found by adding additional clinical factors, looking at changes in the genetic code and some biomarkers, they could approach what a pathologist could do on the order of 98% predictability of who was going to develop metastatic cancer. It turns out 15 of these initial 40 high-risk ones are actually low-risk group, and they would actually have been tremendously over-treated, whereas 10 of the low-risk group actually have a high risk, looking at the whole data set, and they would have been undertreated. The outcomes in medicine will become much more predictive. The implications for this in terms of the original cell lines and their dedication to these processes is also, I think, going to be important in biology. We have to understand the environmental components as well as the genetic ones. We're overemphasizing the genetic ones because we can measure them now. We're starting broadly with the environment. We're seeing if we can apply shotgun sequencing broadly to environmental sites. We started with modestly with the Sargasso Sea. The Sargasso Sea is the only ocean or sea that's bounded by ocean currents. The Gulf Stream comes up the coast here and goes over to Europe. We started off of Bermuda with just essentially a 50-gallon oil drum full of seawater, 200 liters filtered through a series of decreasing size filters, collecting the microbes on a 0.1 micron filter and the viruses on a 50 kilodalton cutoff. The samples, the filters were brought back. This is the Tiger campus in Rockville. This is our new 120,000 square foot building that will be completed in April and house all the institutes. This is the facility where we currently do over 100 million letters of genetic code 24 hours a day, 7 days a week with a small team. What we found was truly stunning. Out of 2 million sequences, we've now discovered over 1.3 million new genes. If you look at the genes and the proteins that are characterized in Swiss prot, it's roughly 138,000, and that includes homologs and paralogs. So in one site in the ocean, we're adding approximately an order of magnitude to GenBank, and these will be going in shortly. In addition, we were able to assemble several complete to nearly complete genomes. The minimum number of species is 1,800. The maximum number is 45,000. The Sargasso Sea is the most nutrient-dilute environment, and we expected to find very low diversity. But in fact, what we found was high diversity, but in ways nobody anticipated. The blue down at the bottom was the entire known universe of bacteria rhodopsins, the light photoreceptors that convert light energy into electrical energy. Off of Monterey, Ed DeLong found one and then went into PCR, and you can see found very similar ones here. We found over 650 new photoreceptors just in this environment in our sequencing samples, going up quite substantially in the number, and we think these may cover every spectrum of the light wavelength. On synthetic genomes, trying to deal with systems biology from a unique vantage point, we're trying to put together genomes from the bottom up. This is Mycoplasma genitalium, the second genome in history that was sequenced, and it has the smallest genome of any free living organism. We started doing transposon mutagenesis to knock out genes to see how many were required for life in Mycoplasma genitalium and pneumoniae, and we found that we could get down to roughly 300 genes. 103 of those 300 genes are of complete unknown function. So here's the most minimal cell we can imagine. All we know is if you knock out one of those genes, it kills the cell. Because these were not serial knockouts, we decided the only way to proceed would be to build a chromosome and see if we could work out exactly, even if it's on an empirical basis, which genes were required. When we started to do this, we found the methods of DNA synthesis completely inadequate for this task and set out on an intermediate project. And 2 days ago, we announced the successful synthesis of Phi-X174 by a new method that allowed us to do it in a couple of weeks. The goal is to build overlapping segments of 5 to 7 genes each that we can stitch together. Deinococcus radiodurans was one of the early genomes we sequenced. This is the radiation-resistant organism that, after having its genome blown apart by as much as 3 million rads of radiation, can stitch it back together precisely the way it was over 12 to 24 hours. We're trying to develop an in vitro recombination system using this that will be able to stitch these large segments together. This paper will be out in PNAS in a couple weeks, but very simply, we just designed overlapping oligonucleotides, had them synthesized. That took the first 5 days. We purified them. They were phosphorylated. Then they were just pooled and ligated with Taq ligase in a high temperature reaction. Then we used a unique approach called a PCA, polymerase cycle assembly, that allows, instead of a PCR, which gives you an amplification, this just gives you an extension and fills in pieces. Then we use PCR to amplify things. Found we could gel purify the amplified chromosomes, circularize them, injected them into E. coli, and ended up with phage. I won't go through the details of PCA, but it's actually very robust, and the pieces that are degenerate get lost in their reaction. I think one of the most surprising aspects of this, instead of adding more DNA to make it better, we found the more dilute reactions favored only the right strands coming together, so you don't get these large branching structures. So this was the purified 5.4 kilodalton DNA that, when it was injected into E. coli, started producing phage particles. This is Phi-X174. I think it's an important historical note. 25 years later, we found only 2 nucleotide possible errors in the Sanger sequence. It's more robust than almost any sequence done to date. But we actually made synthetically the Sanger sequence that was in GenBank, and it led to live virus. So it's possible there were not errors, but subsequent changes in the genome. We have a team now trying to use the new photoreceptors. We're trying to modify photosynthesis to see if we can take light energy directly into hydrogen production as an example. And we see a series of synthetic cells enabling us to do everything from understand the basic components of cellular biology to maybe having some practical applications with pharmaceutical and hydrogen production. I think in the very near future, designed and engineered species will replace much of what we know as the petrochemical industry. DuPont's already starting to do this. They make propane diol now from an extensively modified E. coli, and the E. coli can make the propane diol faster and cheaper than their chemists can. In the future, most sources of food will have these chemical components, possibly sources of energy and bioremediation. Let me give you just a simple example. Taxol, which came from the bark of the yew tree, you'd have to kill about 10 100-year-old trees to treat one breast cancer patient with Taxol. Chemists have not been able to totally synthesize Taxol. They start with another tree now, so they don't kill the yew trees with an intermediate. A genomic approach might be to start with the genome of the yew tree, put together these pathways in a synthetic genome. Possibly in the future, we'll have synthetic multi-cell species as we begin to understand what Lee and others are calling systems biology, perhaps able to restore other species. But our current estimate of the gene pool has gone from what people were thinking was a relatively low number to our current estimates are between 10 and 20 billion genes in all of biology on this planet. When you start to understand that number, we're obviously much better trying to preserve that diversity than trying to recreate it in the future. I figured following Lee, I had to come up with some speculations that would make his speculations look minor. As you know, that's virtually an impossible task. So I'm going to predict that biology and computing will merge. And if you think these predictions looking forward, I always say let's look back 10 years. The Palm Pilot most of you use today has more power than the average desktop from a decade ago. I think the next bullet is most frightening. A Nintendo Game Boy has more compute power than all the computers used to send Apollo 11 to the moon in 1969. Some people argued it was just a computer game. Even more stunning, in 1992, the largest computer was only 59 gigaflops. At Celera, to assemble the human genome, we had to build a 1.5 teraflop computer that was the third largest computer in the world in 1999 and 2000. Today, it's not even in the top 100. The largest today is in Japan, the Earth simulator at 45,000 gigaflops. Moore's Law is going to continue to drive this forward. I had an interesting discussion with Gordon Moore, and I made the argument that Lee did that genomics is moving faster than Moore's Law. And he indicated it obviously can't keep doing that for long because we're going to be dependent on the future of computing, so they have to merge at the same pace in the future. The expectation, if you follow Moore's Law, within 20 years we should have computers that do hundreds of petaops the size of this podium, and think of how that could transform a medicine. So I think Lee and I are in total agreement within a decade we'll have new technology that allows all of our genomes to be sequenced. So we can envision very large databases of tens of millions of genomes where associated with clinical and phenotypic information will be, I think, the only way we will ever resolve the genetic components of human disease and human behavior, except for the rare diseases that are with one or a few genes. Many people working on computer interfaces, even for cognition, and there's been some recent success at Princeton and Columbia. This is obviously a advanced computer chip Here's a diatom whose genetic code is now being sequenced that can lay down single silica tracks much finer than the best computer chip etching. So maybe in the future we will genetically build our computers and have them interface. To really go out on a limb, to quote Stephen Hawking, genetic engineering with human is going to occur whether we like it or not. I don't think it's going to occur in the next decade or so. I just came from a meeting at Rockefeller. I'm planning the next 1,000 years, and they're trying to build their endowment. And so we came up with a prediction, maybe in 300 to 500 years, when we really have this comprehensive understanding of our genetic code, we might begin to modify it. Obviously, it would make sense to try to eliminate the genetic basis of disease if it wasn't eliminating behavior. But I think these potential issues make it even more important to have an intelligent discussion today. Thank you very much.